Welcome to this platform. So in this video, we look at chapter 4, that is biological molecules for the new syllabus IGCSE Biology 0610 for the exams in the year 2026, 2027 and 2028. So if you are writing your exams in these years, this is the material or this is the video that you wouldn't want to miss. At the end of the lesson, we are going to answer multiple choice questions, that is paper 2 questions from extended, and those questions should be able to help us even to understand the topic in detail. And of course, we get exposure on how the questions are phrased. So this is the information that you wouldn't want to miss. All right, now what do we need to know about these biological molecules? Okay, so first thing first, you should know that the bodies of living organisms are made up of many different chemicals. And three main categories of molecules in living organisms or chemicals are carbohydrates. So we have carbohydrates, proteins, and fats. Now, what is very important here, I want you to understand this part very well. So let me put here, we are saying carbohydrate. Let me just in front of carbohydrate. So carbohydrate is energy-giving food anyway. But what is important for you is to know what it is made of. So carbohydrate is made up of carbon, made up of hydrogen, and made up of oxygen. For those that want a mastery thing for mastering this, you can just say CHO. So carbon, hydrogen, and oxygen. These are components of carbohydrate. Now for protein, it's made up of carbon, is made up of hydrogen. So also find hydrogen in proteins. You also find oxygen. But in addition to that, proteins have nitrogen. So very important to understand this. In addition to that, they have nitrogen and in some cases, sulfur. So very important that you understand this. Okay. Then fats, what do they contain? So fats, they contain carbon. Fats, they contain hydrogen. Fats contain oxygen. So in other words, both or all the three contain CHO, but only nitrogen has an addition of only protein has an addition of nitrogen or sulfur in it. So very important that you understand the component of these molecules. Now, because of what they contain, they are also known as organic molecules, and simply because they contain carbon, hydrogen, oxygen. Okay, so very important that you understand the component of this, and that's the first objective that we have achieved. Now, let us look at this one by one. So what can you say about carbohydrates? So we are discussing now those three molecules in detail. So for carbohydrates are large molecules that is made up of simple sugars, otherwise known as monosaccharides. So when you talk of carbohydrates are large molecules that made up of simple sugars known as monosaccharides. Monosaccharides simply means sugars. So sometimes you not find monosaccharides, but you find sugars or glucose. So carbohydrates, they are made up of monosaccharides, which are just sugars. Sugars can be glucose, can be galactose, it can be fructose. So those are sugars in other words. Okay, but very important that in most cases they refer to just sugars or glucose. So glucose and sugar is the mostly used. So very important that you know this. Now, glucose is an example of monosaccharide, as I've already said. Now, when glucose or a lot of glucose molecules, so let me say this is a glucose, a lot of them, they combine, they normally form what we call polysaccharide. Okay, so when a lot of glucose molecule react, they form what we call polysaccharide. But if only two that combine, they form disaccharide. Example of disaccharides, we can talk of maltose is a disaccharide that is made up of two glucose molecules. So only two glucose combining form a disaccharide called maltose. But a lot of glucose molecules, they form what we call the polysaccharides. Now, what is important here is that you understand or you know examples of polysaccharides. So example of polysaccharides or large molecules is starch, glycogen, and cellulose. Now, this is very important. So glucose is a simplest unit from which the polysaccharides such as the glycogen, starch, and the cellulose are made. Now, sometimes you find questions where they ask you, name the polysaccharides, or you are given the diagram where we have the name of the polysaccharide and the basic unit from which it is made. So these glycogen, starch, and cellulose, they are all made up of glucose. So very important. As we look at the questions, we are going to see that this is very vital information. So now, which means starch is made up of a lot of glucose. Now, what is the difference between starch and glycogen? For those that have not gone further to look at digestion, the difference between starch and glycogen is that starch is just the storage form of glucose in plants. So the storage form of glucose in plants is called starch. So starch is found in plants. Then, of course, the storage form of glucose in animal is what we call glycogen. So glycogen is simply the storage form of glucose in animals. But for cellulose, it makes up the cell wall. I guess we've gone through the first part and you understand this part. So this diagram that you are able to see here just demonstrate. So this is a monosaccharide, one monosaccharide joining together forming disaccharide. And the example of disaccharide we said is maltose, not maltase. Remember, maltase, it is an enzyme. So not maltase, but maltose. So very important that you understand this. So this can be a sugar, which is a glucose, then maltose, then a lot of sugars combining, as we can see, forming a polysaccharide, whatever the name is. It can be glycogen, it can be starch or cellulose. I guess you've picked something here. Let us look at fats. What about fats? So these are large molecules made from smaller units of, so each of these molecules, please, very important that you understand what is made up or what makes up these, the basic unit, in other words, the simplest unit. So for the fat, we are saying they are large molecules and the smaller unit of these are fat, acids, and glycerol. So fat, acid, and glycerol, basic units of fats. Okay, so fats can alternatively be referred to as oils. And of course, we are going to look at oils is just simply fats liquid at room temperature. Very important. Now, the basic units is glycerol molecule chemically combined with 3 fat acid. Like we've already said, fat acid and glycerol make up the fats. They are basic units. Now, fats are liquid at room temperature, or they are called oils. So oils are just fats at room temperature and room temperature rather. So the example of the example of a fat molecule is this one that you're able to see. So this yellow part that you're able to see is the glycerol. So this one is the glycerol in the case you find the diagram. Then to it, what is attached is fat acid. Now, we are told 3 fat acid make up 1 fat. So this is also a fat acid and also another fat acid. So in other words, a fat is made up of 1 glycerol and 3 fat acids. Okay, so these are basic units. Very important that you understand this. Let us look at proteins. So proteins are molecules that are made up of long chain of smaller molecules. Now, very important also you understand the basic units here. So the basic units of proteins are amino acids. Okay, the basic units of proteins are what? Amino acids. Now, amino acid, there are about 20 types of amino acids. Okay, sometimes they can be presented in terms of shapes. You find different shapes, circles, you find shapes like triangles, you find shapes like square, you find shapes like rectangle, whatever shapes that you can find, but these represent amino acid. Now, when these amino acids are joined together, they form a large chain, otherwise known as a protein. So a protein is made up from smaller, or it is made up from amino acid, lot of them joining in different pattern. So like I've already said, there are about 20 amino acids. The amino acid can be arranged in any order, resulting into hundreds of thousands of different proteins. So they can be arranged in a random pattern. Different in combination give rise to different proteins. Okay, so very important that you understand this. Now, in this case, the diagrams that you are able to see, amino acids were presented in terms of colors. So different color represent different amino acid. And as you can see, forming a peptide combining in that order. Then, of course, coiling here to form what we call a protein. But what is important you to understand here is that amino acids are made up of proteins are made up of amino acid, and there are about 20 amino acids that combine in a different pattern, resulting into hundreds of proteins. So very important that you understand the basic units of proteins. Then now we have looked at the three molecules. It's very important that now we look at food test. Now, when you talk of food test, is just the test carried out to find out what nutrient or food nutrient the food contain or the sample contain. So there are different tests which we can be carried out to detect the presence of what are the food tests that we test. So we can test for starch, can test for reducing sugars, we can test for proteins, fats and oil, or vitamin C. So these are the food tests that we are going to carry out. So this is very important as you learn this part, also very important as you prepare also for paper 6. So the practical paper, you might find this. So very important that you understand the procedure and also the color changes, the reagents that are used and all that. So let us look at the first one, which is starch. Now, what do you do to test for starch? So in this case, what is used as an example is the potato. So the potato contains starch. Now, you slice the potato or peel the potato, then add iodine solution. Iodine solution is brown in color, or sometimes they will use yellow. So brown in color. Now, what happens when you add it on the potato that contains starch? That is what we are going to look at. So look at the procedure. So add the drops of iodine solution to the food sample. So the food sample, in this case, we are using the potato or Irish potato. If the food sample contains starch, it will change from orange to blue-black. So the color change is very important. So this potato, if you peel it and pour iodine here, if it contains starch, obviously contains starch, it is going to turn blue-black. So the positive indication of starch is blue-black. But of course, if starch is absent, the sample might remain brown, as that is the color of iodine solution. So very important that you understand that. Brown is the color of iodine solution, so it indicates negative result for starch, but blue-black indicate the positive result. Very important as we go through the questions, as we are going to see. Let us look at testing for reducing sugars. So what do you do for testing for reducing sugars? So please, on the first one, very important or things that you should pick here is the reagent that is used to test for starch, which is iodine solution. So iodine test is carried out to find out whether starch is present. And of course, the positive part shows blue-black. Very important. Then when you talk of reducing sugars, the reagent is Benedict solution. So Benedict's solution is the reagent. Now, what is the procedure? Add Benedict solution to a test tube of food sample solution. Now, the amount of Benedict solution that you add and also the sample, they should be equal. Sometimes you can add 2 ml of some Benedict solution to 2 ml of the food sample in the test tube. But what is important also is that here there's heating, and this is the only reaction, or this is the only test that requires you to heat. Very important that you understand that. So one, Benedict solution is used as a reagent, and of course in this case you add equal amount of a sample and the Benedict solution in the test tube containing the sample solution. Then heat. The temperature should be to 60 to 70 degree, and you don't heat direct the test tube, you put it in the water bath, as you can see here. So this contain water bath, then now you put the test tube. You don't heat direct, but sometimes you can heat direct on the burner. Now heat for about how many minutes? 5 minutes. Observe the color change of the solution. Now what will be the color change? Now very important also that you know the color of Benedict solution. So Benedict solution is blue. So when you put it in the test tube containing solution, you expect the solution to turn what? To turn blue. So you expect the solution also to turn blue. Now, when you heat, the presence of reducing sugars is going to give you brown as a color, orange, or red. Okay, so these are the colors that you expect. So here we are saying reducing sugars are present in the food sample. The solution will turn from blue, so the original blue, to orange, brick red, or sometimes yellow, even green, depending with the intensity of the reducing sugars. So long as it has moved from blue, just know that reducing sugars are pleasant. So very important that you know the solution, the procedure, the color change that take place from where to where. These are the questions that we are going to meet. So the setup, just like you are able to see, so Benedict solution here being added to the test tube, taken for heating. Heating, we use the Bunsen burner. Then of course you observe the color change. Let us look at testing for proteins. So for proteins, no heating is required. So add drops of Bullet solution to the sample solution in the test tube. If there is protein in the food sample, the color will change from, so here the color changes from blue to violet or purple. So here also the color of Bullet is blue initially. So under this testing for proteins, you need to know that is Bullet. So the moment you see Bullet, you should relate it to protein testing, because in questions, they will not tell you, they will just ask you which one shows the positive indication of protein. So where there's Bullet and turns purple or violet, that is the positive indication. So the presence of protein in the solution and in the sample food turns the solution from blue to violet or purple. So as you can see here, Bullet reagent added to this turns purple. So this one shows positive. If it remains blue, it shows negative indication. Let us look at testing for fats. So one of the popular testing for fats that we look at is what we call emulsion test. So here is what we call emulsion test. Now in this emulsion test, you use alcohol, specifically ethanol. Now what happened to this? You add ethanol. So look at this, add 2 ml of ethanol to the test tube with food sample and shake well. Pour the ethanol into equal volume of cold distilled water. If the substance or if the test substance contains lipid, a cloud emulsion will be observed. Look at this. So if you carry out that procedure, then now you do whatever you have to do. If a solution remains clear like this one, it means negative. But if it becomes cloudy like this one, it means positive. So what is used to test for fats is ethanol, and positive indication produces cloudy or emulsion form. Okay, so very important that you understand this, and I guess there's no problem up to this point. Let us look at testing for vitamins. So for vitamins testing, you'll use what you call DCPIP, otherwise known as dichlorphenoryndophenol. Okay, so dichlorphenoryndophenol, that is one that we use, otherwise known as DCPIP. Now, DCPIP is blue in color. So this originally is blue in color. Now, what do? Add 1 ml of DCPIP, which is to the test tube. Add some food sample to the solution. If the food sample contains vitamins, the blue color dye of DCPIP will disappear. So in other words, this is what is going to be the thing. So you have the sample here, which is the Minute Maid, orange juice. Then we have, that is DCIP, Rendophenone. Okay. Now, when you add it to this, it is blue in color. So when you add the sample to the DCPIP, it turns colorless. So the color change is from blue to colorless. So very important that you understand the color change and also the reagent that is used. I don't think something that would fail to remember. All right, so we are done with the testing for sample, but there's one more molecule that we need to look at, which is the DNA molecule. So what is important under this one is to look at the structure of the DNA molecule. Now, let us look at it. So the DNA is the molecule that has all the instruction that living organisms need to grow, to reproduce, and function. You already know from where we are coming from, the DNA contains genetic information. Now, each DNA molecule is made up of 2 strands. So very important, each DNA molecule is made up of 2 strands coiled together. Now, coiled together, we can say close linked. This is one of the terms that you need to know. Close linked together to form a double helix. So they are close linked together to form a double helix or helix. Now, each strand contains bases. Again, very important that you understand that each strand contains what? Contain bases. Okay, the bases always pair in the hydrogen bond between pair of bases. Okay, again, another thing, hydrogen bond between pair of bases holds the strand together. The bases always pair in the same way. So what you need to know about the DNA is just the molecule that has all the instruction that living organisms need to grow, reproduce, and function. In other words, it contains genetic information about what is produced in the body. Okay, each DNA molecule is made up of 2 strands coiled together. So 2 strands coiled together or cross-linked to form a double helix. And each strand contains bases. Hydrogen bond between pairs of between bases holds the strand together. Okay, so let us look at the example of this DNA and how it bonds. So as you have here, the nucleotides, you have, but what is important here are the bases. Okay, so please just correct the name here. That is of the bases, just correct the names. So the name is adenine. So here is supposed to be adenine, which is just correct. But this one is supposed to be Y, like that. Then cytosine, which is the just okay. Then guanine. So these are the names. So this is A, this is the T, this is the G, this is the C. But you don't need to know the name. You just need to know the letters C, G, T, A. You just need to know that and which one bonds with which one. So very important that you understand here, or what you need to understand is how they pair. So C pairs with G in the triple bond. Then of course A pairs with T, but you don't need to know about this. All what you need to know is that C pairs with G, A pairs with what? T. Very important. Okay, as you can see here, the pairing. Then of course here we have the sugar, phosphate, and all that as a backbone. This is what we call double helix. So they interlink and form what we call the double helix. So this is the double helix. Now, as we start looking at questions, what is going to be important is the base pairing and also the percentage calculation. What is the percentage of that? What is the percentage of that? So we are going to look at that. All right. So which means this is the last part of the lesson. So please, if you've enjoyed up to this point, do not forget to subscribe and do not forget to invite others. Do not forget also to register for membership so that you can receive videos specifically just for you. And also you can request a topic where you can get to do a topic specifically for you, and also you have to, or you get to benefit from that lesson. All right, so let us look at the questions now. So we are going to look at the questions now. As you can see, IGCSE Biology 0610, that is chapter 4, Biological Molecules, paper 2 exam questions, that is for extended. which is multiple choice from the year 2021 to 2025. So we are going to answer the questions. All right, so this is the first question that we are going to answer, and for this one is coming from paper 2, variant 1, May, June 2025. So the question was on question 5. A DNA molecule contains pair of bases. Yes, we know. What is the correct combination of pair base? What is the correct combination of pair of bases? I said C bond with what? G. Then T bond with A. Okay, it can be AT or it can be TA. It doesn't really matter. Can be G, C or C, G. So long as you know that C bond with G, A bond with T. So which one is correct? A cannot bond with G, C cannot bond with A, G cannot bond with T. So the correct is D. So T and A. What is the test for proteins? So question 6, the same year. What is the test for protein? So name of the test. So the name of the test is Bullate. So we don't care about Benedict and also Benedict there. So it's Bullate. Does it require heating? No. I said only reducing sugars or Benedict requires heating. Then what is the color? Blue to purple. So the answer here is C. Look at how the question comes. Then now let us look at this one, paper 2, variant 3, May, June, that is 2025. In a DNA molecule, bonds between pair of bases hold two strands together. In a DNA molecule, bond between pairs of bases hold the strands together. High temperature can break these bonds to form two single strands. So when they are heated, the bonds can break. The base pair C and G is held together more strongly. You know that C and G is a triple bond. Held together more strongly than A and T. A and T is a double bond. So you expect this one to be strong. But that is not the question. The thing is that two DNA molecules containing the same number of bases were studied. In a DNA molecule 1, 20% bases are T. Now, do you know what it means 20% are T? So look at this. We are saying T pairs with what? Pairs with A. So where you find T, you find also A. If we are saying this is 20%, it means that also this is 20%. So they pair in equal. So 20%, 20%. So out of the total percentage, we already have 40%. So 40% is as for A and T, which means G and C, this one, they share equally. So which means what remains out of 100 is 60. So which means this is 30% and this is 30%, making it 60%. And this gives us 100%. I guess you get that. Now, what is the question? Which DNA molecule would require the lowest temperature to break into single strand? Which one would require lowest temperature to break into single strand? Look at this. DNA molecule 1. Is it DNA molecule 1? DNA molecule 1, say, contain 20% of T, which means it has 30% or more of what? Of G, which are very difficult to break. So this one cannot be. Then DNA molecule 1, because they are more G than, they are saying lowest. So this one is different. The more they are, the pair of G, the more heat is required. Now see, DNA molecule 2, because there are more A bases. Do we have more A bases? Which one will require? So DNA A has more of, no. It's supposed to be DNA 2. Look at this. So for 1, we've calculated, is the one that we've calculated. So for 1, if it's 20% for this, which means G is 30%. So this one is hard to break because we have more of this. But for this second one, if it's 20% of C, which is same as 20% of G, so 20% of this, which means it has A of 30%. So more of the A are easy. So in other words, DNA 2 because there are more of A bases present. So this is the answer. I guess you've gotten something here. Then let us look at this question. So this is paper 2, variant 2, February, March 2025. A section of a chromosome contains 4,500 bases. A section of a chromosome, so a chromosome is just where the DNA is coiled. So contain 4,500 bases. There are 1,400 G bases. Now, these are bases, not pairs. There's a difference between 1,400 pairs, which means pairs it will be C to G, but not pairs. So, which means this is one pair, but this is a base, which means just just G. So there are how many of these? Okay. Then they are saying how many A bases are there. Now, out of the total 4,500, G are these. It means that you also have the same number of C, 1,450. So the total number of these C and G giving us, that is 0, 0. We have this as 9, then I think is 2,900. So it gives us 2,900 for these two. Now we need to find the base of A. So we are going to say 4,500 minus 2,900 is giving me 1,000. So 1,600. Now this 1,600 is for both A and D because we just want for A, but you know that the pair is 80. So just like we did here, if one is 1,400, which means also the other one is 1,400. Now here we want for A. Then we have this for the two bases. So which means we divide this one by 2. And you are going to find 800. So the answer is 800. And I guess you've gotten a thing here. Which chemical element is found in protein but not in carbohydrates? So remember, component of protein, carbohydrate, and fats, CO, but in proteins you have N, which is the nitrogen. So only in protein is nitrogen. Let us look at this one, which is paper 2, variant 1, October, November 2024. Which statement about biological molecule is correct? DNA molecule contain pairs of bases. A pairs with G. No. A pairs with T. So this is wrong. The second one, fat acids and glycerol molecule can be joined to form a protein. No, they form fats. Glucose molecule can be joined to form glucose molecules can be joined together to make cellulose and glycogen. Yes, yes. Starch molecule are made of maltose. Maltase, no, not maltase. If it was maltose, yes, maltase is an enzyme. So the answer here is, you see, glucose molecules. Remember, among the polysaccharides that I talked of, cellulose was one of them. So cellulose, glycogen, starch are polysaccharides made up of glucose. So the answer is C. Then we look at this question, that is paper 2, variant 2, October, November 2024. Which base in a DNA molecule always pair with G? So the one that pairs with G is C. By now, I'm sure you are familiar with this. Let us look at this one. So this is paper 2, variant 3, October, November 2024. Which food testing reagent shows a positive result when it turns from blue to purple? Which food testing reagent shows the positive result when it turns from blue to purple? Look at how clever the questions are asked. So here is bullet. You know that you are testing for proteins. Then let us look at this one. The diagram shows the stick end of the DNA of the plasmid that has been cut by a restriction enzyme. The unpaired bases are labeled. So these are unpaired bases. Now, what is the correct sequence of missing base? So the missing base down here. Now, you need to know that C pairs with G, this one with T, this one with G. We have G there, so this is C, this is A, this is C. So GT, C, A, C, GT. So you're looking for GT, GT, C, A, C. So this is the answer. Question 8. Test was performed on the food substance. A positive result was shown by the color change to blue-black. Remember, blue-black is starch. Which solution was used to test? So they've not asked what kind of or what was being tested, but they've asked us of a solution. As I said, you need to make sure that you know the solution. So in that case is, or in this case is iodine solution. It's the one that is used to test for starch. Let us look at this one. That is paper 2, variant 2, May, June 2024. The diagram shows the bases in the section of the DNA from one strand only. So, from one strand only, this. What is the base sequence of the complementary strand? So, which means the opposite one. So, it's going to be T, G, A, G, T, C, A, G. So do we have TGAA? So we want the one that is starting with T. TC, no. So TGAA, so already is this one. All right, the diagram shows 4 molecules. C represent carbon, H represent hydrogen, O represent oxygen, and N represent nitrogen. Which structure is an amino acid? So we are going to skim through or look through these diagrams and look at the one that has the nitrogen, because amino acid is related to protein. So we are looking for the one that has nitrogen, and it is this one. So B is the amino acid. This is paper 2, variant 2, 2024, that is February, March. Which row shows the element that make up a carbohydrate molecule? So carbohydrate made up of carbon, hydrogen, oxygen. So where there's carbon should be yes, where there's hydrogen should be yes, where there's nitrogen no, then oxygen yes. So this is the answer. Question 11, large molecules are made from smaller ones. Which row names the large molecule and the small molecule it is made from? You remember that I said such questions you find them. So for me to explain, which means I have seen quite a number of questions. So large molecule cellulose made from smaller molecule glucose, yes. Large molecule fat made from glycogen, no. Glycogen, fat acid, no. Starch, amino acid, no. So the answer is A. This is paper 2, variant 1, October, November 2023. What is the color change shown by Benedict solution when heated with reducing sugar? So reducing sugars are present. So Benedict solution is blue. It changes to red or yellow or green or brick red. So blue to purple, no. Blue to red, yes. Brown to blue black, this is iodine. Red to yellow, no. So the answer is B. Paper 2, variant 2, May, June 2023. DNA contain pair of bases. Which pair shows correct combination? So A cannot bond with C. C and G yes. G and A no. T and G no. So the answer is B. Paper 2, variant 2, October, November 2023. The first question was, which row identify the chemical element in protein? So here we are talking of protein. So protein contain carbon, contain hydrogen, oxygen, and nitrogen. So throughout should be ticked, and that is A. The table shows large biological molecules and some small molecules. So large biological molecule, we have cellulose, oils, glycogen, protein. Now small molecules, so we are looking for a corresponding small molecules. So the first one is the answer, that is cellulose, glucose. Oil, amino acid, no. Fat acid and glycerol. Glycogen, and glucose, yes. Then protein and ethanol, no. So it is 1 and 3. Which row correctly pair large molecule with smaller molecule used to make them? So 1 and 3, which is B. This is paper 2, variant 3, October, November 2023. Which row shows the chemical element contained in fats? So fats contain the same as carbohydrate, carbon, hydrogen, oxygen. So shouldn't be nitrogen. So nitrogen should not be ticked. So we are targeting this one or this one. Now the fact that this one is not ticked is not the answer. So the answer is A. Glycogen is a large molecule made from smaller molecules. Which smaller molecule glycogen is made of? So it's made from glucose. The diagram below shows part of the protein. So this is the protein, we've been told, protein molecules. Now, protein molecules is made up of smaller units, these smaller units called amino acids. So these are amino acids. What does X represent? So represent amino acid. Question 8. That is structure of a DNA involve two strands coiled together to form a double helix. Which pairing of bases between the strand is correct? So C cannot bond with T, C cannot bond with A, T can bond, so it's B. That is paper 2, 2023, May, June, that is 2023. Paper 2, variant 3, May, June 2023. The table shows the result of a test carried out on the fruit or on the fruit. So this test was carried out on the fruit. So Benedict solution, you know that Benedict is for sugar, so positive. Bullet for protein, positive. Ethanol for fats, which is ethanol emulsion, negative. So iodine solution, negative. So this food or this fruit had no fats and no starch, but had proteins and sugar. So what did the fruit contain? So protein and reducing sugar. Question 8, describe the structure of the DNA. So how is the structure of the DNA? Each strand coils, or each strand contain chemicals called fat acids. No, each strand contains sequence of amino acid. Each strand contains mRNA. Two strands coil to form a double helix. So two strands coil to form a double helix. But is this also the possible? What makes B the wrong answer? So which one is the answer here? So put up a debate on this part. Let us look at this paper 2, variant 2, February, March 2023. Which substance is used to test for vitamin? So what is used to test for vitamin is DCPIP. Question 8, in the DNA, what is the correct base pairing? So TT, no. TC, no. TG, no. So it's TA. The diagram shows a length of a DNA. So this one is paper 2, variant 1, October, November 2022. What is the sequence of bases in an unparalleled or in an unlabeled strand of DNA? Starting from the top of a diagram. So starting from the top of a diagram, unlabeled part is this one. So what does A pair with? T. What does T pair with? A. What does G pair with? C. C, G. Then this one, T. Then this one, G. So it will be TA. So the one that starts with TA. So not this one, not this one, not this one. So TA, CG, TG. Yeah, so the answer is B. This is paper 2, variant 2, October, November 2022. DNA is a molecule consisting of two linked strands. Part of a single strand of a DNA molecule has the sequence this. So part of it has a sequence of this. What is the sequence of the second strand? So the second strand needs to correspond with this. So on the first part it will be T, then here G, then here G, then here C, then here A, then here C, then T, T. So it should be TGG, CAA, CTT. So the one that starts with it, T. So TGG, that is the AACTT. So the answer is C. This is paper 2, variant 3, October-November 2022. Which statement about the structure of a DNA is correct? DNA contains 6 types of bases? No. DNA contains 3 strands coiled to form a helix? No. 2 strands. DNA is made up of a chain of amino acids. DNA has 2 strands which close link between the base pairs. So this is correct. Question 9. A doctor tested the urine of 4 patients for both, so look at this, glucose and protein. The color of the urine sample after testing are shown in the table. So color of testing for Bullet, so Bullet blue, blue. So the one that remained blue, then here color for testing for Benedict. It cannot turn, brick red. Then this other one remained blue. Now what is the question? Which patient urine contained both protein and glucose? So protein and glucose. So we need purple on the protein, then brick red on this other part. So in other words, it's C. This is paper 2, variant 1, May, June 2022. What are the smallest units or basic units of a starch and glycogen? So starch and glycogen, they are made up of glucose. So we should find glucose, glucose. So the answer is T. In the length of a DNA, 20% of the bases were T. So T, 20%. Now T pairs with A. It means that also A is 20% because these need to be in equal proportion. What is the percentage of G? So G and C, we are going to share the percentage that remains. So this is 40% out of 100. So what remains is 60%. Now 60 divided 2, so which means this is 30%. And also this is 30%. So the answer is 30%. This is paper 2, variant 2, October-November 2022. What are the smallest basic unit of starch and glycogen? So this one is the one that we are from answering. So the answer is D. Then here is a question on question 8. A chromosome was analyzed and found to have 6,000. So 6,205. So let us answer question 8. So for question 8, a chromosome was analyzed and found to have 6,125 base or pairs of bases. Now, if these are pair of bases, you know that pair of bases is a pair, this and that. So there are 2. So which means this one, 1,025 multiplied by 2. Now, if you multiply, what I want is to find the total number of bases. Okay, so these are pairs, but to find the total number of pairs, I mean the bases you multiply by 2. So 6,125, that is 6,125 multiplied by 2. So it's giving me 12,200, please, 12,200, that is the 12 to 250, like this, 12,250. This is the total bases. Now we are told just for G, there were 2,345. Now, to multiply by 2, because these are equal or in equal proportion, if G were 2,345, which means also the corresponding C was this same number. So multiply this number 2,345 multiplied by 2 is giving me, out of these 12,250, 4,600 were bases of C and G. Now, if you subtract this from 12,000, what remains are the bases of A and T. So minus, that is 12,050. I'm finding 7,560. Now, this is total number of A and T. So divide or share that number by 2. Okay, so divide 2 and it's giving me 3,708. So the answer is B. I'm sure you've gotten the explanation here. Then question 9, which type of molecule are enzymes? So enzymes are proteins. So this is the part that we are yet to look at enzymes, but enzymes are proteins. Let us look at paper 2, variant 3, May, June 2022. What are the smallest basic unit of starch and glycogen? So this question has repeated even here. Question 8, DNA is made up of dash strand coiled together. So it's made up of double strand or two strands coiled together. The strand contain chemicals called bases. So it contains chemicals called bases. These chemicals form dash. Okay, so cross link. So we are looking for two, then bases, then let's see, it's made up of two strands, then of bases, not three, two strands of bases cross link. So the answer is B. Let us look at paper 2, variant 2, May, June 2022. Which row is correct for a positive test for vitamins? So vitamins, color of the food test reagent. So DCPIP is blue. So you're going for blue, not colorless. It turns colorless upon adding the vitamins. Heated, no. So no heating. So already know this one has yes heating, so we are going for this one. Then final color of the test reagent, blue. No, it becomes colorless. Okay, so blue, no, colorless. So this is the answer. All right, we've left out one question. In which molecule are close link formed between bases, DNA, to form a double helix? This is paper 2, variant 1, October, November 2021. The bases on one of the strand of a DNA molecule have the sequence. What is the corresponding sequence for the strand? So I'll leave this one to you. I've answered quite a number of questions on this. And of course, I guess this part is very simple now. Then let us look at this one. That is paper 2, variant 1, May, June 2021. This one should be the continuation. Yeah, it's a continuation of what we've not answered. What element is found in protein but not in carbohydrates? Nitrogen. The diagram below shows the short section of a DNA strand, that is the strand of or single strand of a DNA. Which strand of a DNA will combine with this strand to form part of a double helix? So TGGA. TGGA. So TGGA, which is this one. Then paper 2, variant 2, May, June 2021. Which element is found in protein but not in carbohydrate? This one we've answered already, nitrogen. Found in protein but not in carbohydrate. Okay. The sequence of bases present on one strand of a DNA molecule, this one. So these questions I've answered quite a number of them. Which sequence shows the base present on the opposite? So on the opposite, you're going to have TAA, CC. So TAA, CC. So this is paper 2, variant 3, May, June 2021. Which element is found in protein but not in carbohydrate? Same question. The DNA sample was tested to identify its bases. A DNA sample was tested to identify its bases. 35% of the bases in the DNA was G. So G was 35. Now, if G is 35%, also it means that C, which is corresponding to G, is 35%. Now, these two make up what is 70%. So which means what remains 30% is shared A and T. Now, what is the question? Which row shows the percentage of other bases? So we have A, C, T. So this is 35, 15, 15, because we share this into 2. So the answer is B. This is paper 2, variant 2, that is February, March 2021. The bases on one strand of the DNA molecule have the sequence CAGCT. So which means the corresponding is GTGA. C, G, A. Okay, so GT, C, G, A. So this is the answer. And this brings us to the end of the presentation. So please, if you are still here up to this point, do not forget to subscribe and also share with others. This is the detailed information that you need for your exams. As you can see, these questions were taken from the exams and of course the latest exams. So as you write in 2026, that is 2027, 2028, this is what you need. Otherwise, see you in the next one.